Hello everyone. Today I will be demonstrating the process of de novo genome assembly of read sequencing data. First of all, what is de novo genome assembly? In a previous demo, we have seen that read sequencing data can be aligned to a reference genome, and you can identify the different features that are present in the sequencing data by comparing the alignments of the reads to the reference genome. We can observe different insertions, small deletions, matches and mismatches in the read sequencing data as it is aligned to the reference genome. We have also seen how to identify small variants on the basis of the alignments of different reads to the reference genome. We have also used a tool called deep variant to identify variants in the sequencing data. However, we regularly encounter situations where reference genomes do not exist for the organism whose data is being sequenced. Now, what do we mean by a reference genome? A reference genome is simply a set of high quality sequences which are known to exist in the DNA of a model organism. When comparing alignments of reads to these high quality sequences, we are able to identify differences based on our certainty that the high quality sequences are correct. However, when we are exploring non-model organisms, we need to have a method to either construct such high quality sequences or be able to infer high quality sequences from the read sequencing data itself. So, de novo genome assembly involves reconstructing the genome of an organism from its read sequencing data without using a reference genome. It is useful when you are exploring the genetic variations in non-model organisms because reference genomes may not exist for such organisms. It can be very useful in identifying the different genetic diversity that is present within a population for whom a reference genome might already exist. Sometimes complex variants are hard to identify simply based on alignment of read sequencing data, and assembled sequences of reads can give better clarity on the existence of complex variants in a sample. De novo genome assembly can also help in identifying or tracking the process of evolution over time. We will be looking at ONT reads, that is to say Oxford Nanopore technology reads. This is a type of third generation sequencing instrument, and we will be looking at ONT duplex reads. Now, what are Oxford Nanopore technology reads? When DNA is sequenced, it is fragmented into smaller chunks, and each of these chunks are passed through a protein called a nanopore. Current is continuously passed through this nanopore, and the amperage across the nanopore is measured. Based on which molecule of the DNA is passing through the nanopore at any given instant, we are able to infer the molecule based on the current that is observed. ONT simplex reads are observed when one of the two strands of DNA pass through the nanopore, and ONT duplex reads are obtained when first one of the strands passes through the nanopore and then the reverse strand passes through the nanopore. The advantage of assembling sequencing data is that the advantage of using Oxford Nanopore technology reads for sequencing for genome assembly is that ONT reads are long, and longer reads will guarantee higher contiguity in the assembly. The fact that ONT reads offer real time sequencing can allow the users of the instrument to decide whether a particular read needs to be sequenced or not. We will not be discussing this much further. However, it is important to note that ONT reads are on average much longer than second generation sequencing data and offer and are useful for generating high quality assemblies. We will be using a software called Hi-Fi ASM for generating genome assemblies. Hi-Fi ASM is a string graph based assembler for long read sequencing data. It is capable of generating phased assemblies, that is to say, if you provide sequencing data of a diploid organism, that is to say an organism which has two parents, or more precisely 2 copies of each chromosome in its nucleus, then Hi-Fi ASM is able to produce phased assemblies, that is to say it is capable of producing 2 sets of contigs which are guaranteed to be part of the same set of chromosomes. It is also an end-to-end solution, that is to say you pass sequencing data in the form of a FASTA or FASTQ file to Hi-Fi ASM and it generates an assembly and outputs the assembly file. There are several different types of assemblers available for users, some of which are listed in this table. You can see that Hi-Fi ASM is a highly accurate assembler. It is comparatively fast and uses a moderate amount of memory and is also capable of generating phased contigs. Hi-Fi ASM was used to generate the first telomere to telomere assembly of the CHM13 sample, and more recently a software called Verco was used to generate the first telomere to telomere assembly of the diploid HG002 sample. So let us look at how we run Hi-Fi ASM. Here I have sequencing data from the first 10 million positions of chromosome 18. It is ONT duplex sequencing data, and we can just take a look at the statistics for these reads. We can see that it is in FASTQ format. It has around 12,000 sequences. The shortest sequence is 512 characters long and the longest sequence is 123,000 characters long. The average length is around 27,000 and the median length, which is the N50 value. The N50 is a statistic which is used to describe the length of the shortest read at half the total length of reads, assuming that you have sorted all the reads in order of decreasing length. So now that we have these statistics, let us assemble these reads. I have already generated one assembly of these reads over here, and I will show you how to run the assembly using Hi-Fi ASM. The command used to generate the assembly has these components. we provide the name of the program -asm. We provide a prefix for the outputs generated by -asm, that is passed as an input to this -o flag. So, over here I have used the prefix asm. For our purposes I will use the prefix test. -t tells -asm how many threads are to be used. In this case we are using 32. The last thing to be provided to -asm is the set of input reads. In this case we are providing the FASTQ reads in the gzipped format. So, we will do the same thing here. The set of reads that I have are stored in the parent folder to the current folder in the gzipped file reads.chr18.10m.fastq.gz. We will store the log in a file called logs, and we can follow it in a separate screen. Okay, so you can see here that -asm first generates a K-mer diagram. This diagram actually shows us how many K-mers there are, how many unique K-mers there are of a specific value. This diagram shows us that there are 10 K-mers, that there are 39,840 K-mers which occur exactly 10 times. Similarly, there are 390,389 K-mers which occur exactly 28 times. This diagram is used to infer the homozygous coverage and heterozygous coverage of the read sequencing data. -asm error corrects these reads 3 times by default, and each time it computes this K-mer diagram to identify the heterozygosity and homozygosity rate. Once it has corrected the reads 3 times, it identifies the homozygosity and heterozygosity rate one last time, and then proceeds with generating the overlaps between all pairs of reads. Once it has identified all the overlaps, it will then create a raw unitic graph. The raw unitic graph contains the information about overlaps between the suffix prefix overlaps between pairs of reads, which are now stored in the unitig format. This graph is further processed to generate a processed unitig graph, and once the processed unitig graph has been created, it is capable of inferring a primary assembly from it. The primary assembly is stored in the p_ctg.gfa file, and it generates 2 more files. Each of these is a partially phased Contig graph of haplotype 1 and haplotype 2. Once these graphs are generated, -asm ends, and we are able to see now the outputs of -asm. So, I have said here that -asm generates GFA files as output. What are GFA files? GFA stands for Graphical Fragment Assembly Format. this is a way of storing the graphs that are generated by Hi-Fi ASM, and each line in this file is tab delimited. A line can start with S or it can start with L, and there are a few other options that these lines can start with. S stands for segment. Immediately after the segment is the ID assigned to that segment, and following that is the content. So if we imagine that we have a read which is being stored as a segment, the read ID would be stored over here and the content of that read would be stored over here. Following the content of the read, the GFA format provides the option for the user to store any additional information. However, the first 3 columns are required. Once we have stored the segments, we also need to store the edges between these segments. These are stored in the link lines indicated by L here. The link lines contain information about the edges between these segments. You store the segment ID, the orientation of the segment ID to which the edge is being drawn, and the orientation of that segment, and any additional information about potentially the length of the overlap or something along those lines. Hi-Fi ASM will also store near each segment a line which starts with A, which contains information about the different reads which are used to create that particular contig or unitig. So let us, now that we have all of these graphs, let us see how we can visualize them. The tool that we will use to visualize these graphs is called Bandage. Bandage stands for Bioinformatic Application for navigating de novo assembly graphs easily. Bandage allows us to process a wide variety of graphical formats. We will only be looking at GFA, and the process is very simple. Let us open Bandage and load a graph. So first of all, what we will do is load these graphs. We will store these graphs in our system, and then we will load them one by one into Bandage. I will be using the GFA files that I have generated beforehand. So I am first loading the raw unitig graph. I have loaded the graph, but you can see that nothing is visible just yet. However, the information about the number of nodes, edges, and the total length has been loaded into the graph. To visualize the graph, we have to click the Draw Graph button, and you can see that Bandage has loaded the graph onto the screen. You can move any part of this graph by clicking on a node and moving it around. If you want to move a specific portion of the node, hold Control and move a specific portion of the node around. You can select multiple nodes by dragging and selecting multiple nodes, and you can move multiple nodes by selecting the nodes first and then clicking and dragging the nodes around. You can see that once you have selected multiple nodes, all of these nodes show up over here. The edges also show up over here. If you want to increase the width of the nodes, you can increase it over here in graph display, and you can choose specific colors based on different options provided by Bandage. You can give lengths to the node labels. However, you can see that this is a bit of a clutter in a graph that is this big. However, if you choose to zoom in, you can either you can press plus to zoom in and you can scroll using your mouse, and it is helpful to have the node labels sometimes, the depth of coverage sometimes, or the length of each node. Okay, so we have visualized the raw unitic graph. Once we have visualized it, well, we may want to save our work, right. One easy way to do that is to click on file and select save image. You can choose to save the entire screen, or you can choose to save just the current view. Now I have already visualized several such graphs and I will go through each of them one by one. Here is the raw unitic graph. Now the first time you visualize a graph, you may see one particular layout. Once you load the GFA file a second time and click on draw graph, you may not necessarily get a graph that looks the same. However, rest assured that the graph is actually the same and all the vertices and edges are exactly the same as any other time you click on the draw graph button. You can see that the raw unitic graph now has two components, and one point is that from this raw unitic graph the assembler needs to infer two sets of contigs, one corresponding to the chromosome 18 from the father and one corresponding to the chromosome 18 from the mother. So let us look at the processed unitic graph. In the processed unitic graph, we can visualize these side by side. You can see that the processed unitic graph is considerably simpler, and this is because several small bubbles that might exist in the raw unitic graph have been removed by Hi-Fi ASM based on the coverage of the reads at each of those locations. From this simplified graph, Hi-Fi ASM can infer a primary assembly, and the primary assembly you can see is one single continuous stretch. Let us load the primary assembly in Bandage. The primary assembly is available in the file labeled p_ctg noseq.gfa. It contains one node and the total length is 9.9 million. Of course, what this means is that it has output one single contig. Let us also visualize the assembly graphs that it has generated for haplotype 1 and haplotype 2. For haplotype 1, we see that there are 3 nodes and the total length is 10 million and 6195. There are 2 components which are incredibly small, and we can just click on length and you can see that they are just 35,000 and 55,000 characters long, and we have a 9.9 million character long contig of chromosome 18. This corresponds well with the idea that we selected reads from the first 10 million locations of chromosome 18. Let us also visualize the assembly graph of haplotype 2, and you can see that now there are 2 nodes. One of these is 8 million characters long and the other is 1.9 million characters long. So we now know how to visualize these assembly graphs using Bandage. Now that we have got a graph which has the assembled data, well, how do we know that what we have got is good? Well, the way in which we evaluate assembly quality is we compute metrics like N50, NG50, the total assembly length, the number of contigs and the longest contig length. The And I have already explained what N50 is for read sequencing data. When we are looking at contigs assembled by an assembler, the N50 is simply the length of the shortest contig at half the total assembly length. So once again, if you arrange all contigs in the order of decreasing length, select the that contig whose length is half, select that contig such that if you add up all the lengths of the contigs before it, we reach half the assembly length. NG50 is the same except that we use half the estimated genome length or the reference genome length. NG50 makes the assemblies comparable across different samples. The total assembly length is an indicator as to whether we have got the correct assembly or not. The total length of the contigs should be roughly equal to the total length of the contigs in a reference genome. If this is not right, that means that something has gone wrong in the way we have inferred contigs. The number of contigs is simply a measure of how discontinuous your assembly is. The larger this number, the more discontiguous your assembly. Of course, this number should not be smaller than the total number of chromosomes present in the sample, in a cell of the sample. The longest contig length is again a metric that says how contiguous your assembly is. The longer this value, the better. QUAST is a tool which you can use to estimate all of these metrics, and we will see how. Before we use QUAST, we already have the contigs stored in the file the contigs for haplotype 1 are stored in this file and the contigs for haplotype 2 are stored in this file. However, this file also contains information about the edges and the links that are present in between these contigs. Now, we don't want those, we don't need that information, and the input to QUAST has to be in FASTA format. So we will convert these GFA files into FASTA, and the way we will do that is by using awk. So over here I have an awk command, and I will pass the GFA file corresponding to the contigs from the first haplotype, and I will store them as just to go through this command. What I am doing is looking at all the lines which start with S, that is to say they are segment lines, and I will print the greater than character, which in a FASTA file indicates that it is a header line. I will print the second column, which is the identifier for the segment, and I will print the third column, which is the content of that segment. Okay. So because I copied these commands, the characters are incorrect. Let me try that again. I see the problem. quotes were incorrect. Right. We will do the same thing for the contigs from haplotype 2. Let us take a look at the contigs that we have obtained. So, we see a contig labeled H1TG1L. Let us look for another line which starts with the same. Okay, so there are two contigs here labeled H1TG1L and H1TG2L. Similarly, for haplotype 2 we have two such contigs. Okay, now that we have obtained these contigs, we can use our old method for visualizing them, but before visualizing these contigs using IGV, let us see how we can use QUAST to obtain the statistics that we have discussed. So, the program is called QUAST.py, it is a Python program, and as an input to QUAST we pass the following information. Specifically, we are passing information about how many threads QUAST uses. We have to provide an output directory for the assembly. We will provide test.hap1.stats. The estimated reference size is intended as an input for estimating the NG50 value, and in our case we know that it is 10 million and we will provide the same, and we will provide the option --large to tell QUAST that we are looking at a large genome, and after this we simply have to pass the FASTA files containing the contigs, and you can see that QUAST has generated test.hap1.stats. We will look at the stats generated for the previous assembly, the assembly that I have pre-generated. So, QUAST generates these files as an output. We can look at the report.txt file to see what output QUAST has generated. So, let us look at report.txt. You can see that there are 3 contigs over here in total. The total length is 10,006,195. The number of contigs again is 3. The length of the longest contig is 9.9 million, and the N50 is 9.9 million. The NG50 is also 9.9 million. We will see a similar result for the contigs from haplotype 2. We see that there are 2 contigs. The total length is 9.9 million. The longest contig is 8 million characters long. The N50 and NG50 are both 8 million again. Okay, so we have seen how to obtain all of these quantities using QUAST. Can we visualize them? Well, yes. Recall that in a previous demonstration we have seen how to visualize the alignments of read sequencing data to a reference genome. Similarly, we can align the contigs that are generated by these assemblies to a reference genome and visualize those. The command for visualizing these is we use minimap2 once again, and we want to generate a SAM file. So, we say -a. We use a preset ASM5 which tells minimap2 that the contigs and the reference are both from the same species, and then output into a file aln.sam. We will convert the output, the SAM output, into a BAM file, sort it and index it, and then visualize it using IGV. Let us look at a visualization. So, the top track over here contains the alignments of the 10 duplex reads sampled from chromosome 18 in the first 10 million bases of chromosome 18. Here is a contig from the first haplotype, and here is a contig from the second haplotype, and you can see that the indels which are, some indels are not preserved in the assembly, and indels which are more likely to be present in the assembly are actually preserved by the assembler. Okay. So, we now know how to visualize these assemblies using IGV as well. Is there any other way to visualize these assemblies? Yes, there is a tool called Sapphire, and you can access it here. You can see that this comes preloaded with some alignments. We can load our own alignment data. However, creating the input for this format is not very straightforward. However, if you have a BAM file, you can click this link to see how to generate the alignment data, which can be used by this tool Sapphire for visualization. So, for now I will load the alignment data. So, let us look at the alignments of contigs from haplotype 1 for Sapphire. I will note that this is not the assembly of the reads from the first 10 million bases of chromosome 18. This is actually an assembly of the entire ONT duplex data for the HG002 sample. Okay, so something has loaded up. It is still not clear what it is. So, here we have the target reference CHM13V1.1, and we can select a specific chromosome that we want to look at. So, let us look at chromosome 18. So, you can see now chromosome 18 there are 2, 3, 4, 8 such contigs available. Let us look at this one. So, this is a contig which is aligned to the first 17 million or so locations of chromosome 18 on CHM13. You can see here, when we zoom in simply by scrolling or pinching, depending upon whether you are using a mouse or a trackpad, you can zoom in simply by pinching or scrolling, and you can move the mouse around to look at different regions which are closely aligned. You can see here that some regions on this contig align to a different location on CHM13, and this is actually an example of how contigs from a sample that is not the reference genome need not actually have arisen from the same location as the reference. So, this is one such contig. Let us look at another contig, right. This is aligned somewhere in the 45 million to 80 million range, and once again you can zoom in to see the regions which are closely aligned. You can click and drag the track to look at the different regions as well. Let us look at chromosome 11. Chromosome 11 looks a little more interesting. There are only 3 such contigs here in the assembly, and let us say we look at the first one, right. So, this region here looks a little bit interesting. If we zoom in, we can see that there are some regions which are in blue, and this one region is in yellow. What actually has happened here is that all of these regions which are in blue are oriented in the same way as they are in chromosome 11 of CHM13. However, this region has inverted. The alignment over here to the alignment over here, this region has actually inverted in our sample as compared to CHM13. You can see over here a far more complicated region where the alignments to CHM13 are not actually contiguous. It probably indicates that there is some major difference in our sample as compared to CHM13 in this region of chromosome 11. We can actually visualize every single such contig once we have got the assembly using this tool. Okay. So, to summarize, we have looked at how to generate an assembly of genome sequencing data, read sequencing data. We have looked at how to generate summary statistics for the assemblies that we have created. We have visualized the graphs that are generated by HiFi ASM, and finally we have also looked at how to visualize the contigs as an alignment to a reference genome. De novo genome assembly helps us understand the diversity of genes and DNA in a population, and because we are assembling reads together, it actually gives us more context when identifying the variants which are present in complex regions of the genome. It is a tool which is a step towards personalized medicine. Thank you.